So it's 10:30. Welcome everybody. It's really great to see so many here. And of course, this is a presidential lecture. And as a presidential lecture, we are really happy to have somebody here that can inspire us the most and have a good story through life, and also have the focus on really great science, going beyond and outside of the scientific knowledge that is common. So I think all this, we have today a very good example, and I think the inspiration here to our interdisciplinary group of people, because OIST is very much about enabling interdisciplinary science, and also in the basic science, which is of course not every place in academia that you can do that. So I think you also will be very interested in really talking to many of the people here, as they will be, and take the opportunity really to, when you get inspired about this presentation here and have discussions, and maybe this will also lead to some collaborations more in the future. And I really want to say that this type of opportunity that the presidential lectures give, well, I want to say to everybody that if you have other possibility thoughts about others that can come and inspire, please let me know, because we would like to really make these type of lectures something that really comes now and then over. This is the second one this year. And so I hope that this will, as I said, catalyze the interdisciplinary interactions. And with that, I don't want to take more time, but I want to give the word over to you. Gerald, you are the host here today. So why don't you take over and give a presentation of the Michael Stryker that we will hear from today.
I'm very happy to welcome Professor Michael Stryker from UCSF. So Professor Stryker is one of the founders of the neuroscience program at UCSF. But he has not only done that. As I learned yesterday, he was like so intimately involved in like the establishment of the new campus of UCSF, like just like OIST, which was a new campus. He even designed the infrastructure of the buildings of this. So the faculty were so involved. Some of the core faculty actually designed the infrastructure that led into this. To me, that's absolutely amazing that the faculty will step up and take charge and make sure that things would be done correctly when, like, for example, contracts are coming. Anyway, so Professor Stryker has had a long and distinguished career, and his training started at University of Michigan, and where and he actually got the degree in philosophy and mathematics, and then from there he went to MIT, where he has PhD with Peter Schiller, and after that he went and did his postdoc with Huber and Weasel, and he was part of the last people who actually did his postdoc with Huber and Weasel when they actually had a joint lab, which is, as many of you know, Huber and Weasel probably basically got a Nobel Prize for vision science, and that's the area in which Professor Stryker works in. After that, he went on to UCSF where as an assistant professor, and pretty much has stayed there since. Other than that, he has, during sabbatical, interestingly, he also had a sabbatical in Pisa, in Italy, where he actually gave a series of lectures to the faculty. And actually, he was, let me get this right. He was the Galileo Professor of Science, and he gave a lecture in a lecture room that was last renovated in 1547. So anyway, this is a little bit newer here, and the City Brenner lecture room. So without further ado, I'll let Professor Stryker take over here.
Thank you, Gerald. Thank you, Gerald. So it's wonderful to be here at last. Many years ago, Sidney Brenner told me, you have to visit this new creation. And later, Torsten Wiesel told me, oh, you have to come to OIST. And fortunately, an opportunity arose, and I'm really blown away by what amazing, beautiful buildings, and what a brilliant arrangement of education you guys have created to bring the students here, to allow them to go to any lab they want because the labs don't have to support the students financially, and to bring together a really diverse set of disciplines that can inspire one another. So I'm extremely grateful to be here. You know, this is just not only physically, but I think intellectually, it seems to be just the way educational and research institutions should be. So today I want to talk about the general topic of why our brains are more flexible when we're younger. But I also want to relate this to the current fad in artificial intelligence and in neural networks, because as the younger of you probably don't know, artificial neural networks, convolutional neural networks, were really inspired by the work of Hubel and Wiesel, who constructed a conceptual model which was verified to some extent of a cascading set of inputs from retina to the lateral geniculate nucleus to the primary visual cortex, the input layer, which had a simple kind of cell, to the successive layers that had complex cells and so on. And this vision of how the cortex worked really was the inspiration for Fukushima's the first convolutional neural network, which is, you know, this is what Facebook uses to recognize your cat. And it was modeled after Hubel and Wiesel's conception of how the cortex worked. And it was the inspiration for essentially all the work that followed and follows in a big way in places like OpenAI and Microsoft and so on. So the idea is that there are inputs that proceed through successive stages which are connected in some specific way to accomplish interesting tasks like what Fukushima did, used recognition of kanji characters and other things. But in those days, most of what the work in neurobiology, given the techniques available, didn't work at the level of individual cells whose connections were hypothesized but not actually really known. But instead, they worked on these macroscopic structures of columns that Hubel and Wiesel discovered, that all of the neurons in a cortical column through the layers shared a preference for responding to a particular orientation, and there was a greater or lesser evidence for a greater or lesser dominance of one eye or the other eye. And these were macroscopic things. The spacing of the columns was on the order of a millimeter. And so it was changes in those columns were the actual data that were measured in many of the experiments. And so the classic experiment in juvenile plasticity was that these columns, that during a critical period in early life, if an animal has normal vision, the columns are more or less equal, the ones driven by the left eye or the right eye. But if during a critical period, the vision of one eye was blurred or occluded, that eye became functionally disconnected from the upper layers and lower layers of the cortex, and the columns of input serving that eye from the lateral geniculate nucleus became shrunk down. And of course, this is a cartoon, but I filled the two eyes with different color label. And, you know, this is a flattened piece of cat visual cortex, which is, you know, bears a close resemblance to the cartoon here, which shows the arrangement in layer 4. And the classic picture from Hubel and Wiesel of the changes in the input to the cortex from the two eyes. They only labeled one eye here, so the white areas are input from the labeled eye. This is the normal situation, and this is the situation if an animal's vision is occluded during early life. And we looked at this a long time ago, and what we saw is what corresponded to those shrunken columns was inputs from the lateral geniculate nucleus, which are like, you know, fully grown hedges that became very sparse, and that took about a week. And this process of plasticity was competitive in that if you closed both eyes or if you blocked activity with tetrodotoxin, you didn't see these changes. So blocking activity didn't cause both eyes to do this or closing both eyes. So somehow there was some interaction that mattered. Now, one of the things we discovered was that the work that Hubel and Wiesel did was focused on the input layer of cortex. But if you looked above the input layer, in layer 3, the next stage of cortical processing, and scored individual neurons on the basis of whether they were driven by one eye or the other eye, or with varying degrees by both, with the middle character being fully equally binocular, what you saw was closing this eye caused this distribution to shift really dramatically within 24 to 48 hours. So it was a much more rapid thing than the anatomical change in the input layer. And the real test of competition was to induce strabismus, where the two eyes point in different directions, or to close one eye one day and the other eye the next day, which had similar effects. And so what this did was create equal amounts of activity from the two eyes, but they were never active driving the same cortical cells at the same time, the left eye and the right eye. And what that resulted was not only a change in the distribution of responses, so there were very few cells driven by both eyes and most cells driven by one eye or the other eye, but there was a millimeter scale rearrangement of anatomical connections between areas of cortex. So these little red dots show the positions of cell bodies that were labeled from these X's. And the normal case is that it's not very selective. You're as likely to make a connection to an area that's dominated by the other eye as you are by the same eye. But after 48 hours in this case, or even 24 hours, these connections are dramatically rearranged. So all this gave rise to the idea that there was competition. And one of the things I want to mention is one often has a good idea for an explanation of what's going on in neurobiology. It's very hard to turn a good idea down. People really strive to accept it. And so, but even if it's wrong. And so this competitive nature of the interaction between the two eyes gave rise to the question, what are the inputs serving the two eyes competing for? And around that time, neurotrophic factors were discovered in the cortex. And so people proposed an analogy to earlier work in the peripheral nervous system and the autonomic nervous system, that the inputs serving the two eyes were competing for limiting quantities of neurotrophin, particularly brain-derived neurotrophic factor, which is one of the ligands of the TrkB neurotrophin receptor. And so the way to test this was to supply extra neurotrophin. So if there was enough for the inputs from both eyes, you wouldn't have any competition anymore. Or the other way to test it would be to block the receptor for the neurotrophins so they couldn't compete on the basis of this. The alternative test turned out to be infeasible. People did experiments that they interpreted that way, but they were actually they were a pure artifact. So, but supplying excess ligand for the TrkB neurotrophin receptor was straightforward. And when that was done, the monocular visual deprivation, and this was done in cats and some of it was done in ferrets, showed a big change from the control distribution to inputs serving responses to the deprived eye were much reduced. And if NT45, which is a less sticky ligand that activates the TrkB receptor in the same way that brain-derived neurotrophic factor, if this was delivered to the cortex, you completely block the effect of monocular deprivation. So boy, we thought we'd struck gold and figured it out. And then even if you lost the responses to the deprived eye, if you delivered this neurotrophin, you could bring the responses back. So from this controlled, deprived animal, you'd lost the responses, but after a period of neurotrophin infusion, they came back. However, one of the things that we saw when we looked more carefully at these cells is in the control animals, the responses were all very specific. They responded to a particular direction of motion or a particular orientation. Whereas in the neurotrophin-treated animals, not only did they lose their selectivity for the eye that had been open, but they lost all their selectivity for responses, and they just responded to anything. And when we looked closer, what we saw was bizarre cells, hairy cells with spines coming from their somata and proximal dendrites, a huge proliferation of what looked like anatomical inputs to these cells in contrast to normal cells. So our conclusion was that this experiment of activating the TrkB receptor did in fact stimulate the growth of new connections, as you might have imagined from work in vitro, but it had nothing to do with normal plasticity. So the idea that they were competing for neurotrophins really did not seem to be supported by these experiments. But these experiments turned up a terrible problem, which is in those days, we did not have genetically modified mice. And I just thought it was hopeless, really, to understand what were the mechanisms of juvenile plasticity, of critical period plasticity. And fortunately, mice came to the rescue. So mice do have a binocular visual field. They have regions of the cortex, some of which are driven only by one eye, some of which are driven by both eyes. And what became clear in a bunch of experiments that I don't have time to talk about is if you closed one eye, deprived vision in one eye, the normal distribution, which in the mouse favors the contralateral eye more than the ipsilateral eye, shifts. And this phenomenon has a well-defined critical period, just as the effect of monocular deprivation in monkeys or cats or ferrets do. And it's associated, we later showed with two-photon imaging longitudinally, it's associated with the loss of many of the spines that serve the deprived eye. Now, mice don't have this beautiful columnar architecture that the animals, the cats and monkeys that had previously been studied have. Instead, it's more of a salt and pepper arrangement of eye preference and orientation preference. But as far as we know, the very same mechanisms that operate here on the macro scale, that operate here and give rise to this macro scale organization, operate the very same way on the micro scale in the mouse. So with genetic access, one could really do a serious test of the mechanisms that are responsible for this competitive interaction in the critical period. And one of the approaches was to use a Shokot inhibitor, where our colleague Kevon Shokot altered the tract B receptor, the receptor for the neurotrophin, so that it was no longer susceptible to blockade by very effective small molecule ligands. But you could chemically make a ligand which would specifically block the altered receptor. So this gave us a way of both spatially and temporally controlling signaling from this neurotrophin pathway. And when we did this, what we saw was that blocking tract B receptors, and we demonstrated that they were blocked, had absolutely no effect on the loss of response to the deprived eye or on the change in the balance between two eyes when we gave the ligand that blocked the neurotrophin receptor. And so this really showed that tract B kinase, this explanation that they're competing for neurotrophins cannot be correct. However, I mean, that was a negative experiment, and fortunately, it turned out neurotrophins do play a critical role. They play a role in the recovery of responses at the end of the critical period. So if you deprive one eye for 4 or 5 days, the cortex loses most of its response to that eye. But if you then open the deprived eye during the critical period, the responses come back, and you essentially recreate the normal situation. And this restoration of responses was due to neurotrophin signaling, so that the expression of the activation of these tract B receptors was absolutely required for the recovery of responsiveness to the deprived eye. And this was consistent with the idea that this recovery was mediated by the rapid growth within 24 hours of new connections. And it had been shown in vivo by work that Barbara, or later Ben Barris, did with Louis Reichert in vitro, that neurotrophin signaling of brain-derived neurotrophic factor for many neurons was absolutely crucial for making new synapses. And in vivo, what you see is shortly after opening the deprived eye, in 6 hours, there's a big squirt of newly synthesized, mature brain-derived neurotrophic factor, and responses haven't recovered at that point. But this presence of the brain-derived neurotrophic factor, which is needed to make new connections in vitro, indeed is associated with, it precedes the response. So our story was then that neurotrophin signaling stimulates or is permissive for the growth of new connections, and maybe more than permissive, and that allows the recovery from visual deprivation. Now, the other aspect of competition involves a completely separate mechanism of homeostatic synaptic scaling. So Rob Malenka's lab discovered in hippocampal cultures in vitro that if you block activity in a chunk of brain for a day or two, and then take that chunk of brain out, the individual synaptic connections become much more powerful. So something about the regulation of synaptic strength depends on a history of activity within the last 24 to 48 hours. And that this effect requires signaling by tumor necrosis factor alpha. So if you look at a TNF, tumor necrosis factor mutant, this homeostatic synaptic scaling just doesn't happen. Well, it turns out that a good test of the competition hypothesis is to look at the monocular region of the visual cortex, because the monocular region of the visual cortex only gets input from one eye, so there's nobody to compete with. And it was long believed that there was no plasticity there, and this was taken as evidence for the competitive hypothesis. So if you record the relative amplitude of the response to one eye, and we did this by a couple different techniques. This was intrinsic signal imaging, and then do monocular deprivation for a bit, close one eye, the eye that provides input to this monocular region, you hardly see any change at all. However, if you block homeostatic synaptic scaling, however, if you look much earlier, what you see is the input to this, the responses in this monocular region go way down, just as they do in the binocular region where there is supposedly competition. But we think homeostatic synaptic scaling brings the responses back. And indeed, if you block homeostatic synaptic scaling with tumor necrosis factor alpha mutants, what you see is the decline of the response is the same, but it just continues to stay the same or decline slightly further. So this is, I think, another nail in the coffin of a mechanism which is competitive. So we have these two mechanisms. They are not the only two, but the normal process of plasticity, which is really the basis of a huge literature on the visual cortex, if you look at it carefully in the mice, the first thing that happens is the deprived eye, the responses get much smaller in the deprived eye. Then as deprivation continues, they stop getting smaller and actually get a little bit bigger, and this was complete mystery. But if you look in the eye that was not closed, what you see is a very large increase in responses. And it turns out this part requires signaling by tumor necrosis factor alpha. Then if you open the deprived eye, what you see is that responses to the formerly deprived eye come back to normal. And during this time, responses in the other eye come down because as the cortex becomes more active, the homeostatic synaptic scaling isn't done as much. So our hypothesis, and this was verified to a considerable extent by the longitudinal two-photon imaging, is this initial effect is not just something like LTP, it's actually the loss of input from the closed eye circuit. Then that's followed by homeostatic synaptic scaling. And then if the deprivation ends during the critical period, there's a regrowth of synapses in the deprived eye circuit. So it gave us a much fuller explanation for what's going on in the juvenile critical period. But part of the point here is that competition, which was the inspiration for a lot of thinking about how the development and plasticity of the cortex work, competition at a macro scale during the critical period is implemented by at least two distinct mechanisms, neither of which is in itself competitive. And so this is a case where a beautiful idea is actually the way at a superficial level, that's what happens. But the actual mechanisms that implement it are not what you would think they would be. So you can also ask the question, why does the visual cortex have a critical period at all? Why is this period of really dramatic plasticity during early life? And it turns out that when the cortex develops early on, the individual cells in the visual cortex become selective by virtue of the way the connections are made for edges at a particular orientation. But the two eyes aren't connected to each other. They don't know about each other. So they each drive cortical cells independently at the whatever random preferred orientation happens to develop. But when we look at the world through our left eye or the right eye, it looks the same. And that's because the binocular cells in our visual cortex are all have similar properties depending, it doesn't matter which eye you drive them through. And that depends on simultaneous vision in the two eyes, binocular vision, during this critical period. So the critical period is the period when the matching of the properties of inputs from the two eyes to cells in the visual cortex takes place. And if you deprive the animals of vision in one or both eyes during the critical period, you preserve this state where the two eyes are driven differently. So we can look then, this is the first third of the talk about juvenile plasticity and how it's implemented. But if we look at adult plasticity, you can say, well, what's going on using this visual cortex model system for adult plasticity? And Huell and Wiesel's answer was nothing, that adults don't have this kind of plasticity. There's nothing like what goes on, they thought, in the juvenile animal. And the evidence for this was you could take an animal after the critical period, a cat, or they did it with monkeys too, close one eye for in some cases for years, open that eye, and you saw that the deprived eye drove the cortex perfectly well. And whereas, in a week or less during the critical period, that eye would become functionally disconnected. And the anatomical changes that I showed also don't happen in adults. In adult mice, there is a sort of form of plasticity with monocular visual deprivation, but it's not a loss of response to the deprived eye. What happens instead is there's somewhat of an increase in response to the eye that had remained open. And this adult form of plasticity, we believe, is part of an attenuated but continued operation of homeostatic synaptic scaling. So if you close one eye, the cortex isn't driven as well. Homeostatic synaptic scaling, TNF-alpha dependent, comes into effect and somewhat increases the response to the eye that remained open. But in addition, if you infuse TrkB ligands into the adult cortex, they don't cause these promiscuous connections or the loss of response specificity. So that while it is true that if you turn off signaling from brain-derived neurotrophic factor, after several months, the cortical cells shrink down. So they do depend on it to some extent, but there's no form of rapid plasticity. And so these aren't the only two mechanisms that are responsible for the change between the juvenile animal and the adult. There are other things like cortical myelination, which also interfere with plasticity in the adult. And I think the first real evidence that at least many people in neurobiology took seriously for dramatic form of adult plasticity, besides the fact that you're all sitting in this room and we always knew there was plasticity in the hippocampal system and with declarative memory. But plasticity that one could understand the anatomy and the circuitry of it, like what we studied in the visual cortex, didn't seem to exist in the adult. And these mechanisms were no longer operative. But my colleague Mike Merzenek worked on the cochlear prosthesis, which is a device with a little microphone in it that stimulates the eighth nerve in an attempt to mimic if the hair cells that signal sound pressure and in the ear, if those hair cells have died, but the nerves that come from the cochlea to the brain are intact, which is true in the most common forms of deafness. The idea was you would try to stimulate those nerves in a way that mimicked what the activity in those nerves during reception of normal sound in a person whose hair cells were intact. And it became clear that biophysically this simply was not possible with electrical stimulation. The activity that was produced in the eighth nerve was nothing like what would actually be there. And so people were depressed about the possibility of a working cochlear prosthesis. But the fact was that if the patients who were given these and allowed to use them for a few weeks or a month, refused to give them back. They found that they could hear, they could understand language, and indeed, the cochlear prosthesis has been a tremendously successful clinical intervention in the clinic from neuroscience. Indeed, one of my children had a 4th grade teacher who was profoundly deaf and only heard by virtue of her cochlear prosthesis. So they can really work. So something was going on then in allowing the people who had cochlear prostheses to interpret this very unnatural form of input to the brain into language. But nobody understood the anatomical rearrangements, how this happened in the brain. It wasn't clear. And Merzenich and I and some others tried to figure out what was going on with this adult plasticity, where there was the anatomical substrate of it wasn't at all clear. And it became clear, at least from studies in experimental animals, that the plasticity absolutely depended on the animals really paying attention. And if you were stimulating things and it didn't matter to the animals and they didn't pay attention to it, there was no adult plasticity. But if you devised things to make them pay attention, then there was. So studies of adult plasticity really required longitudinal experiments in alert animals. And we adopted a procedure of having a mouse run on a styrofoam ball suspended in air where we could hold its head fixed, but the mouse was free to run around wherever he wanted and would behave pretty normally in many respects. And this procedure let us study the activity of the visual cortex while the mouse was either engaged or not engaged. And the thing we discovered, then this is work primarily by Chris Neal, a postdoc in the lab who, a fantastic scientist who has his own lab now, was that there was this very substantial change in cortical responses as soon as the animal started to walk or run. And it didn't only happen when he walked or run, but walking or running turned on this system and put the cortex into a high gain state. And if you looked in the lateral geniculate nucleus, there was no such change. So this was a turning up the gain of the operation of the visual cortex that was really pretty dramatic. And we wondered, what was the neural circuit that implemented this? And so one approach was to start with walking or running. And so what we did was to stimulate the midbrain locomotor center. And if you look at the blue light, as soon as that turns on, the mouse starts to run. As soon as it turns off, it doesn't. And of course, when it turns on like this and the mouse starts to run, the cortex goes into this high gain state. Well, this experiment's completely uninformative because it doesn't matter whether you're, making him run with a blue light or whether he runs just by himself, the cortex will go into a high gain state. What was informative, however, was that we could turn down the intensity or the frequency of this light and make the cortex go in the high gain state without inducing locomotion. So this said it's not the descending connections that make the legs move that's important for engaging the cortex in the high gain state. It's really something having to do with the ascending inputs from this area, which there are several of them, and it became hard to follow them all, and I'm not going to go into the details of that. So the other thing we did then was start at the top instead of starting at the bottom and looking in the visual cortex to see were there any cells that received this signal for locomotion. And in the mouse, there were a large number of different classes of neurons to which we had genetic access. And it turned out that a small population of inhibitory interneurons that express vasoactive intestinal peptide, the VIP cells, their activity paralleled that of locomotion. So, and that was kind of surprising because they're inhibitory, and yet when they're active, the cortex responds more strongly. But actually, what the circuit that was worked out by Massimo Scanziani's laboratory showed that the VIP cells activate somatostatin cells, and the somatostatin cells inhibit the cortical neurons. So when VIP cells are active, they turn off a population of somatostatin cells and allow the excitatory cells in the cortex to respond much more strongly. And we believe that is the explanation for why the cortex goes into this high gain state. And if you optogenetically activate the VIP cells in sedentary mice, you can put the cortex into the high gain state. If you photolytically damage them, or later there were other techniques, we could compromise release by using genetically, you removed the ability of locomotion to put the cortex into the high gain state. So the animal could walk or run, but it didn't make any difference to the cortex. So the question was, does having the cortex go into this high gain state, which we think is normally produced by locomotion or by attention, does that enhance plasticity in the cortex? And yes, indeed, if you use a mouse model of human amblyopia, what happens is that responses, if there is a droopy eyelid during early life or a cataract that gets removed, what happens is that responses to the deprived eye gradually get better, but they only get about halfway to the normal level. But if you give visual stimulation during locomotion, the responses get all the way to the normal level. So we had sort of three stories that running enhances cortical responses. We had the subcortical and cortical circuit that is responsible for that, and that running enhances plasticity, presumably through the enhanced responses. But does it enhance plasticity directly without running? Or is there aerobic exercise or something that produces some signal? So to answer this, we used a tetanus toxin that we expressed in VIP neurons to keep them from being active. And so there is no enhancement of activity during locomotion, and no change in their selectivity for stimulus orientation. But silencing the activity of these VIP cells completely blocks the plasticity that's gated by locomotion. And activating them, and I'm sorry I'm not explaining all of these axes, but activating the VIP cells really permits this adult plasticity without locomotion. But there was something really strange about this, because the plasticity we saw was only an enhanced response to the stimulus that the animals viewed during locomotion. So if they viewed a noise pattern during locomotion, the response to the noise pattern was enhanced, and enhanced for weeks. Or if they viewed just a single bar or a grating, their response to that was enhanced, but the response to the noise pattern or to anything else was not enhanced. And so this enhancement of response absolutely required locomotion in our apparatus. And when we looked at individual cells, and in this experiment, we, during a period of locomotion, we stimulated the visual field either with 45-degree lines or 135-degree lines. And what we saw, looking at the same cell after before and after a week of such stimulation, that if the response of the cell was activated by the stimulus that was shown during locomotion, it got much bigger. And, but if the response of the cell was not activated by the stimulus that was shown by locomotion, it didn't change. And so this shows a stimulus-specific enhancement of visual responses. So this is a form of adult plasticity which operates cell by cell and keeps and regulates the degree of responsiveness of neurons. So I think our conclusion is that there's this rapid and persistent increase in visual cortical responses to stimuli that are presented during locomotion. And I stress it's not only locomotion that puts the cortex in this state, but locomotion was a wonderful tool for us in that we could reliably turn it on. And the enhancement was evident using completely non-invasive methods, including intrinsic signal imaging, where you don't have to put anything in. You can do it through an intact skull and just looking at a change in light absorption, as well as with 2-photon calcium imaging or other techniques. And the increase in response, this form of plasticity really depended on how well the neurons responded to the stimulus beforehand. So what's special about the high gain state that produces plasticity? Is it a useful thing to the animals? And we think it is because when the cortex is in the high gain state, there's much more information in the visual response. So if we look at a mouse that's in our, on our styrofoam ball, looking at visual stimuli, different visual stimuli, and we're recording a bunch of neurons, 40 to 100 neurons simultaneously, we can ask, how well from the activity of these neurons can you tell which stimulus is present? So essentially, you're decoding the activity of the visual cortex, presumably maybe like the way the brain does. And so you can measure how many bits of information is each cell contributing to this process. And if you look at an animal that's running, in the first 100 milliseconds of response, it gets a certain amount of information. More information is a lower value here. And if the animal is stationary, you need about 300 milliseconds to get the same amount of information. And it's not just because there are more spikes here, but there's more information in each individual spike. So this system is designed when the cortex is in the high gain state to create informative responses much better. And those are exactly what you would want if you're going to do plasticity. We know that this requires the VIP cells in our hypothetical circuit. I mean, it's a real circuit, is that VIP cells inhibit the somatostatin cells, and relieve the excitatory cells of inhibitory input. But the VIP cells also secrete this peptide, and peptides are associated with plasticity, slow-acting plasticity in the brain. So we ask the question, which is it? Is it the GABA, the inhibitory neurotransmitter that the VIP cells release, or is it the VIP itself, the peptide? And it turned out that the plasticity was completely normal if we knocked out the peptide, and it was completely gone if we attenuated inhibitory signaling. So it's really a signal of activity in the brain. And I persuaded my younger colleagues to do this experiment to ask what happens if you look at an old animal. Does this, this plasticity is a feature of adult mice, but if you look at a really old mouse, is it still there? And it was initially discouraging because it only got about halfway there in older mice with the normal amount of induction. But if you just kept doing it longer and longer, twice as long, you got almost as much plasticity as you would get in a young mouse, but it didn't last very long. So it's not a very good message for people my age. But so, these what I've been talking about is a mouse, but these anatomical elements of the VIP cell circuit for enhancing gain are present in all of us, in all these animals that have been studied. They depend on, and I didn't show you the anatomical evidence first, they depend on cholinergic signaling through the basal forebrain, through the horizontal limb of the diagonal band of Broca, which I'm sure none of you want to remember. But I think we believe that this same circuit that's activated by locomotion accounts for, or may account for, selective attention and the enhanced learning that it produces. And it's an interesting prospect to try to exploit this for therapy, and it actually is being used now in a study at the Mass General Hospital that I'm not involved in, but I was very glad to see. So we would like to restore juvenile plasticity in adults. And we don't really know how to do it. And I'm going to have to skip over a lot of this because I talked more slowly than I did when I rehearsed this talk. But we know that cortical plasticity is gated by interneurons. And that comes from much older experiments we did where if we make interneurons secrete BDNF more, if we make their maturation a week earlier, the critical period becomes a week earlier. If we delay their maturation by 2 weeks, the critical period is 2 weeks later. So we decided to take, in a collaboration between Arturo Alvarez-Bulla's lab and my lab, we decided to take embryonic inhibitory neurons and transplant them into postnatal cortex and see whether it was possible that they could induce a second critical period. So this is the normal critical period. The interneurons are born, around embryonic day 13. The normal critical period happens, peaks around postnatal day 28 or 30, 33 to 35 days after the inhibitory neurons are born. So if we transplanted them shortly after birth, what we saw was a second critical period that was very similar to the first. It had a well-defined duration. And if we transplanted them even later, we could get a second critical period later. So we had 2 critical periods of juvenile-style plasticity. So we were thrilled with this. And in fact, there's a company, Neurona, which is trying to take advantage of this fact to restore plasticity clinically in adults. I'm not associated with that company, but I was glad to see it, although it's a tough row to hoe for them. So it turns out that it's only the interneurons from the medial ganglionic eminence, which are parvalbumin cells or and somatostatin cells, that are capable of doing this. And if it turned out that either one of them alone could do it, but if we killed off both the parvalbumin and the somatostatin cells, then we got no plasticity. Then we could not induce a second critical period. And this depended, just as for the VIP cell story I told, the induction of the second critical period depended on the release of GABA from these transplanted interneurons. So they would grow and mature and differentiate, and they were just fine. And none of the other neurons, even if we transplanted them, they were happy to differentiate into their normal selves, but they did not induce the second critical period. So the one thing we worried about was, did these transplanted interneurons really induce a second critical period? Or were they just creating their own separate circuit that inhibited responses from the deprived eye? And that wasn't a crazy thing to think. And we were inspired by Christian Levelt's lab in the Netherlands, had shown that if you measure the responses to the 2 eyes and you silence these inhibitory neurons, for 5 seconds while you measure the response without their activity or measure the response with their activity, what you see is the responses change, but the relative balance of inputs from the 2 eyes does not change. So one of my postdoctoral fellows, Mahmoud Hosseini, did the very same experiment with the second critical period. And what we showed was that turning off the transplanted interneurons did not change the ocular dominance. So if we had created a second critical period and got the host circuitry to respond differently, it still responded differently to the two eyes when we turned off the transplanted interneurons. So this transplantation is a reorganization of the host circuitry, and I really wish we knew what went on, how this works, and we're actively working on this with genomics techniques and with some modeling approaches that I don't have time to describe. But in the last 3 minutes, I'd like to address the question that's, I think, a very interesting one. Given the origin of convolutional neural networks in the visual cortex, how similar is the visual cortex to the convolutional neural networks that are so popular these days? Is the visual cortex really working like them? And this is a collaboration between my friend Steve Zucker, a mathematician at Yale, and Greg Field, who's a retinal neurobiologist who's now at UCLA. And you know, in the old days, Hubel and Wiesel studied neurons one at a time. But we can study lots of neurons at once with electrodes that record from, 50 or 100 of them or more. And it's possible to do this in the retina. So how have people dealt with this huge amount of data from simultaneous recording that is possible to obtain now? And essentially, the approach has been and we use this to look at information transmission, to decode the simultaneous activity of a bunch of neurons in order to figure out what stimulus drove the neurons. And so the approach has been to construct a manifold of responses in a higher dimensional space, of which this is a sort of pseudo 3D projection. And each dot is a single trial. And what you see is that the dots for the trials for a particular stimulus tend to be clustered close together on this manifold. So this is a decoding manifold, and it's a very widespread approach in neuroscience for dealing with high-dimensional data. We were more interested in asking about how the neurons are connected and how they respond in the circuit. So instead of trying to decode the response, what we tried to do was an analysis which said, how similar are the different neurons when you use a whole ensemble of different stimuli to activate them, and constructed a manifold so that each dot is a neuron rather than each dot being a stimulus. And the neurons that are closer to one another have more similar responses. And so this is a neural encoding manifold. And the idea is that if you have a network, you can calculate what the neural encoding manifold from that network should be, and that where nearby neurons would respond similar to similar stimuli. And depending on how this manifold is arranged, you can then do the inverse problem and try to figure out what's the network that gave rise to this manifold. And it's an ill-posed problem mathematically, but we, in simple cases, our mathematics colleagues solved this problem. And essentially what you need to do is express neurons as a function of their responses to stimuli, so you have to reduce the dimensionality, and then you have to infer the manifold from the similarity of responses. And so this is an example of the manifold from retinal responses to a whole ensemble of stimuli. This is, a 3D picture of it. This is a 2D projection. And what you see in the retinal manifold are all these clusters where there are lots of neurons that are very similar to one another distributed in the manifold. And it turns out each of these clusters is a particular retinal ganglion cell type. And you can tell this by the tiling of their receptive fields in in visual space. So the manifold in the retina looks like what you would expect it to look. In the visual cortex, however, the manifold we see is completely continuous. It isn't clustered like that. And if you look at it and draw a geodesic on the manifold, you can see gradual changes in the responses. These are post-stimulus histograms where the hotter colors make stronger responses to different stimuli that we used. And there's a geodesic in the other direction, the way the responses change. But the cortical manifold is really quite different from the retinal one. And if you have a network with a bunch of cells that are not connected to one another, that results in a manifold which isn't continuous. If you have partially connected networks, then you get a manifold that's a little bit connected. And if you have a densely connected network, the manifold ends up being continuous like this. So we were interested, if you take the deep convolutional neural networks like VGC or ResNet 50 or any of the popular ones where the code is available to use, you can say, how do these networks treat our ensemble of visual stimuli? Do they do it like the visual cortex, or do they do it differently? And it turns out, and there's lots more figures that would show this. They're tightly clustered. So essentially, the convolutional neural networks are not like the mouse cortical network. They're more like a big retina than a small brain. And it's an interesting thing. It will be very interesting to see whether when we look at monkeys and animals whose visual systems are more like humans, whether the cortical networks really are very distinct from these computer networks that do certain things very well. So this is a list of the people who've been in my lab, and this is a very strange talk to give. Usually you just talk about one experiment with all the details and the controls, but I tried to give an overview to answer some bigger questions. And most of the people on this picture and our collaborators from a couple different labs have contributed to different things, and I hope I mentioned the names when I was talking. Thank you.
So I was a bit intrigued by what you mentioned on activating VIP neurons could enhance cortical activity, given that they are predominantly inhibitory. So yes, their role should be suppressing whatever rather than enhancing. But on the other hand, there's some evidence now that these VIP neurons, in addition to releasing GABA, could also release acetylcholine. Mm-hmm. So could they actually be enhancing locomotion by releasing acetylcholine? Although there's a small population of VIP neurons in the cortex that could be doing this.
Yeah. So we were looking in the visual cortex, and the situation may not be the same everywhere else, but activity in the visual cortex doesn't do anything to locomotion. You can, animals walk or run perfectly fine if the visual cortex is gone. So the signal that comes from the locomotor center gets to the cortex via the basal forebrain cholinergic neurons, which activate the VIP cells. But VIP cells are also activated by top-down inputs from frontal areas of the cortex that presumably are involved in attention and higher cortical function. So, it makes sense that because the major output of these VIP neurons is to these very powerful somatostatin inhibitory neurons, that when the VIP cells are active, the somatostatin cells will be shut off, and that would allow the same input to generate a much bigger response in the cortical cells. So
I see. So it's to disinhibition.
Yeah, it's through inhibition of another inhibitory pathway. The VIP cells that we studied were all the same. They all did the same thing. The somatostatin cells were only about between a half and a third of them had activity that was the opposite of what the VIP cells did. So there are other kinds of somatostatin cells that participate in other circuits. But yeah.
Thank you so much for this amazing talk.
Loud.
Oh, thank you so much for this amazing talk. I have many questions, but I'll just ask one. On the last part about comparing the visual cortex or the visual system up to that point versus the deep convolutional network, and your conclusion based on this data is really interesting. But I wondered, in the ResNet, for example, we know how we are training the system. What if, for example, the different result that you get in the visual cortical neurons is just purely explained by the fact that we may not know what is a training signal. So if for example, the network structure or the basic layout is okay, but what is being used to train is different, can we distinguish such a case?
Yeah, it's an interesting question, but we were looking at a visual cortex in an adult animal that's already trained up. Whatever experience the mouse has is what trained the network. And of course, not all of there's a whole story about making cortical maps and the origin of these cortical circuits that Dave Feldheim at the University of California, Santa Cruz and I worked on, showing that Ephrin A signaling and in combination with neural activity was what set up the maps in the visual cortex and in the superior colliculus, what determined connections between them. So I think we don't, I'm quite confident that the mouse visual cortex is not organized to make individual cells look like the individual units at any one of many stages we looked at 3 different convolutional neural networks that were the popular ones for vision, and they all were pretty similar and all different from what the mouse visual cortex is. The question in my mind, which I hope we'll have an answer to in a year or so, is whether if we look at a monkey that has a visual system that's much more like the human than the mouse visual system is, will the monkey resemble the convolutional neural networks? There is fragmentary evidence. There's reason to believe on both sides from what's in the literature. And nobody else has made these manifolds the way we do that really shows the relationship of neural encoding. So the question is I don't know, but I don't think we're looking at a system that's already trained up and organized the way it is. So, yep.
Yeah, thank you very much. Coming back to the effect on locomotion, there's something more simple as environmental enrichment, for instance.
Yes, and my friend Sigurd Löwel, who's in Göttingen now, and who came to my lab to start working on mice. so she has shown that if you raise mice in a much richer environment, an environment that's probably much more like the natural world, that the juvenile plasticity lasts for much longer. You don't have this 1 week critical period, but you can get a juvenile form of plasticity lasting for at least months. It's a very, but what they do, they have like a 5-level condominium where the mice, and there's both social stimulation, the food is hidden in a different place every other day. So the mice really have to be engaged and pay attention and be active in their environment. And in Sigrid's experiments, she's shown very compellingly that you can preserve the juvenile form of plasticity much, much longer, and who knows, maybe indefinitely, although she hasn't been able to do it in old mice. But this is very labor-intensive, hiding the food every other day. So, yeah, I mean, it's, and Takao Hench's lab has shown that there is a molecule which Mike Greenberg's lab identified as being important for the differentiation of inhibitory neurons, that essentially you can delete it or suppress it later in life and restore some degree of juvenile plasticity too. So there are more factors than the ones I talked about, and I think I think the environmental enrichment is really wonderful, and it suggests that the time course of these things that we see in lab-reared mice may really depend on animals that are not exerting the, they're not in their natural environment where they get a much greater amount of activity and stimulation. It's just, and this is a big problem for us who work in a lab, because there are very few experiments that have been done like this of animals in a natural environment. And I think it's a crucial thing to do. And I'm very glad that some of the younger people are doing these experiments now, because I don't like to say that we've been working on an artifact all our career. And it's very clear, even with humans, normal humans and monkeys, that there is a critical period. But what limits the critical period, like the best data on the human critical period for the visual cortex is from a Japanese surgeon, Awaya, who plotted the impairment of vision that came from cataracts at different periods of life in human babies. And then the cataracts were fixed surgically, and then and so what was clear is the first 6 weeks postnatally were by far the most devastating. And there was less and less sensitivity, but there was still considerable sensitivity that lasts until 6 months in humans. So, but in monkeys, Hubel and Weasel studies of monkeys that you can ask, they at least had some social environment. They still had a critical period which had a very clear dramatic ending. So I don't think we're just studying an artifact, but of cages. Yeah.
Thank you very much. On the first part, the developmental plasticity, if the account is that to get correct binocular vision, you have to have the same view from both eyes aligned to align the perceptive field properties of the neurons. With the mice, when they're most of the time doing monocular vision, or each eye is moving around independently, how does that work at all?
No, that's not true at all.
They don't.
Mice have very conjugate eye movements. So if you measure the movements of the two eyes, there are vergence movements where they, but no, the eyes move. They go like this. They're very closely yoked, so they're really. In the binocular part of the visual field, they're seeing the same thing at the same time.
So what do you think about, I know rats, they can move their eyes independently, and I know some birds. So with these animals that don't do it?
There are some animals that do it, and actually, a long time ago, I was really interested in working on, the flatfish, where one eye migrates from the bottom side to the top side of the fish. because clearly they don't have simultaneous vision, but they don't have binocular cells either. So yeah, and rats, I haven't studied the rats, but I think rats probably have mostly conjugate eye movements as well. Yeah, so I don't think that's an issue. And you know, you don't have to do any surgery. You can just fit, these are experiments that were done with cats and with chickens. You can fit prisms over the eye that just deviate the optical axis with no surgery whatsoever, and you get the same effect as creating strabismus and moving one eye. So, yeah.
I have one more question. You said something extremely fundamental and interesting about a good idea, not quite being true in terms of implementation. I recently heard a really very interesting talk by Florian Engert, who obviously can image from all of the neurons in a zebrafish brain. And there was a decision-making sort of task or reflex that a zebrafish was doing, and they could fit this state, drift diffusion model, and come up with some parts of the circuit that may be implementing recurrent part, maybe some part might be implementing a decision sort of threshold, for example. Yes. And so they could find these neurons. Do you think that just finding neurons that kind of correspond to your model is a valid confirmation, or this is quite philosophical, I guess, but I find it very interesting.
It's a good question, and those of us who work in mammals have to be envious of the kind of work that Florian is doing in zebrafish and Misha Ahrens and others, or that Corey Bardman's lab is doing in C. elegans, where you know all the neurons and you know what they're doing. On the other hand, I think our results can be very strong because we can do loss of function and gain of function acutely, and we can turn off the signaling at a well-defined time or turn it back on, and or we can stimulate them extrinsically, and I think get strong evidence that our idea about the role of a particular neuron in a circuit is the one that's really there. But I think your general point is, early in my career, I was completely seduced by an idea about how the visual cortex developed. And I had made computer models of it, and the idea that the organization of the visual cortex and the development of orientation selectivity depended on the visual environment. And there were, they were experiments, artifactual experiments had made it into every textbook, showing that if you raised kittens in a painted cylinder with vertical stripes, the only neurons that were present in the visual cortex were selective for vertical. And if you looked at animals that had never had visual experience and you studied their neurons, what you saw is they weren't selective for orientation at all. And if you put them in one of these environments, they became selective for orientation. This was in the day when you recorded from an individual neuron at the end of a sharp wire and studied its activity and assessed its selectivity. You judged it, you just judge it by trying different things and listening to the response of the neurons and writing down on your sheet of paper what its selectivity is. And some people, some groups got it absolutely right, everything they published. And Hubel and Wiesel did. And that's largely because they would never agree with each other. And so they, I did experiments with them. And when you were studying one of these neurons, you had to convince both of them. And that was really hard. So, but you can imagine that this technique was subject to wishful thinking. So in the end, I had tried to extend these experiments about how the development of the brain depends on the visual environment, and failed to be able to extend them. And so finally, and fortunately I was this graduate student at MIT at that time, so there were resources, like there were big things of electronics components that, so we, in order to do these experiments properly, we created, my PhD advisor and I, he for a different purpose and I for these plasticity and development experiments, created the world's first computer-driven optical display so that you could present randomly interleaved stimuli and actually measure the responses to everything. It's hard for people to believe now, but in those days you couldn't put images on a video screen from a computer. You had to make a very fancy optic bench, basically a very fancy slide projector, to project these images to study the neurons and get objective data, how they were responding. And the result was that the very first neuron we recorded from an animal whose mother had been in the dark for the 2 weeks before birth, and where the animals remained in the dark until the 3rd week of life when they're physiologically stable enough, but they'd never seen anything in their whole life. And the very first neuron we saw was perfectly orientation selective. And so that made the plasticity experiments even more exciting because, even if you didn't require visual experience for development, seeing the neurons only be one. And it turned out that result was wrong too. That what happened was that the neurons that were innately selective for some orientation, if they never saw that orientation, they became poorly responsive and non-selective. So if so what happens is most of the neurons, if the animal only saw one thing, were just ignored by the people who did it. But what had happened to those neurons is they just stopped responding very well, and people ignored them. So there was an effect, but it was not what was advertised. So this beautiful idea that I'd made all these computer models of, and lots of other people had done it, about how the development of this specificity depended on experience was complete bullshit, basically. But there's no substitute for getting good, objective data. And we get seduced by ideas, and we're often reluctant to give them up. But this, it's science. We didn't. If we actually knew the answer, we wouldn't have to do the experiment. So I think this is I can point out like 4 instances in which there was a beautiful idea that turned out not to be right. But I tell the people in my lab, if they do an experiment and the result is contrary to what they expected and what they hoped for, I said, it's not your fault. We didn't make it. We just work here. Our job is to find out how the world is, not is it what we want to be. And it makes me worry about a lot of the current scientific literature, where a lot of it seems to be a sort of high-pressure sales job for something rather than a clear, a clear presentation of all the reasons on the various sides for something. So anyway, that's a life lesson. So.
Thank you very much for the wonderful talk. I'm actually machine learning, so I have a question for the CNN part, following the first questions. So you said that CNN is not the visual cortex.
Not the mouse visual
not the mouse
cortex.
Visual. Okay. So this part is very interesting, and because there's lots of clusters.
Yeah.
Right. So, but in machine learning point of view, we try to make the learning to make the clusters. So this sounds, after the learning, we try to make the clusters clearly. So
Yeah.
this sounds a little bit contradiction to what we are doing as a machine learning. So do you have
Yeah.
any suggestion for us to improve the learning system?
That's a very good point. So we didn't know, and we are only beginning to know now, what's going on with the matching of binocular inputs to the visual cortex, because the real data for that requires following. Is it the case that you have a cell that's selected for this in one eye and this in the other eye, and say the stronger one dominates and gradually they get closer and closer together or something like that? And the data that would answer that question has to come from recording the same cell over time and seeing what happens to it. There is no substitute for longitudinal data in a development question. And even though it's hard to get, and my Josh Trachtenberg at UCLA has done some experiments which really suggest that it's not what we thought at all, that when you have cells that respond, say individual cells that respond to two different orientations at the beginning, what happens is they just become monocular cells and they lose one input. And the new cells you see later that are binocular are cells that were formerly monocular and acquire a response in the other eye. So it's the process is completely different from what everybody had envisioned and made computational models of. So that's okay. I mean, so there's only this one experiment, and more needs to be done on this problem. But I think it's another case where the process that you imagined was operative in development seems not to be. And it's a different process that's more selective. It's a selectivity and enhancement of something rather than a change in the circuit. But there's really only this one paper on that that's very recent. So I think other labs and other approaches will look at this. So
Thank you. Thank you.
yeah.
Let's thank Professor Stryker for this wonderful talk.
Thank you.